individual neurons from that region of the brain, the SCN, that's the clock that controls your 24-hour activity, and you can put them into culture as Dave Welsh has done here in our lab, and you can measure in real time transcription occurring on a daily basis inside of these cultured cells. And what's really amazing that you could see from this movie is all of those individual cells, when they're cultured, are cycling differently from all the other ones. So there must be something in vivo that collects these cells together and makes a concerted rhythm that controls our sleep-wake cycle. If we dissociate these cells, their little clocks all run happily along individually, but in the living organism, they must be built together into a tissue that actually beats out a pacemaker for life. And what we've learned from these types of experiments using luciferase is lots of individual tissues have their own clocks, and they receive orchestrating or entraining signals from the pacemaker that's inside the SCN. The SCN is a master pacemaker for receiving light signals. Like I said, they receive those signals from the retina, and they entrain the rest of the oscillators in the organism. So the story now in 2007 is now much more complicated. And don't worry about the gene names. Just think about this. We've learned recently that clocks are not made up of one feedback loop. They're made up of multiple feedback loops. These feedback loops mesh with each other in the same way that wheels of gears mess with each other. And this elaborated architecture appears to make a much stronger, a much more robust clock than one single loop can. And what we've learned from science is that this architecture, again, is conserved evolutionary between mammals and flies, even though some of the different proteins are used. So that's a view inside of animals.